Everybody, today we're going to be talking about topic 3.4, inheritance. So this might be a refresher from 10th grade, where we talked about pedigrees and Punnett squares and genetics a little bit. So hopefully you can see the connections to that, as well as where we've just come from talking about meiosis and how that leads to our sex cells and our new combination of alleles. All right, some traits that you may or may not know about that are genetic and inherited from your parents or from your grandparents is a widow's peak. So the widow's peak is actually a dominant trait, and having a straight hairline is a recessive trait. So if you only have one allele to have a widow's peak, you will have it. You will express that in your phenotype. Another trait is tongue rolling. Who would have thought that this wasn't just a cool skill that you learned on your own? It's something that was passed on to your parents. Tongue rolling is also a dominant trait. So one of your parents will be able to do the tongue roll. Dimples. Dimples is a dominant trait as well. So only one of your parents has to have had the trait for dimples in their genetic code, and you will have it. You will express it. The attached earlobe. So when I say attached earlobe, I'm talking about this earlobe over here. You can see this is called a free earlobe, where it's loose on this part of your ear. This is considered a dominant to have a free earlobe and recessive to have a attached earlobe. And this is me. I definitely have attached earlobes. So sometimes they're called heart ears, elf ears in a way, but they're called an attached earlobe and they're a recessive trait. So let's review some of these terms that we learned in 10th grade when we talked about genetics before, because they're going to pop up again. So whenever we're talking about someone's genotype, we're talking about their actual genes, so the DNA that they have for a trait. So this would refer to the allele that's on one of their chromosomes, or remember in their homologous pair, they're going to have an allele on each chromosome for a trait. That's their genotype, okay? The phenotype is actually the characteristic of an organism. So it's what we see. It's what the person's expressing. So it's what the blue eyes. I know someone has blue eyes. That's their phenotype. But I don't know their genotype right away just by looking at them. Homozygous versus heterozygous. So this is the way that we talk about what the allele combination of a gene is. So whenever someone says someone is homozygous dominant or homozygous recessive, that means that they have both the same letter or the same allele, we would say. So two identicals allele for a gene. So if it's homozygous recessive, then they have two of the recessive alleles. If it's homozygous dominant, then they have two of the dominant sized alleles. Hetero means different. So heterozygous is referring to two different alleles. So one of the dominant alleles and one of the recessives. Or sometimes we might say one uppercase letter, one lowercase letter when we're looking at the alleles in a Punnett square. So bringing back those words dominant and recessive, remember dominant is an allele that has the same effect on the phenotype whether it is present in the homozygous or heterozygous state. So what that means is even if you have only one dominant allele in the heterozygous state, this is what you're going to express. For the recessive allele, it only has the effect on the phenotype when it's present in the homozygous state. So you have to have both of the recessive alleles in order to express it. And sometimes we might refer to it as two little letters. So here's an example below. So if we're talking about the trait or the gene for earlobe attachment, let's say the alleles are represented by a big F for the dominant and little f, lowercase f, for the recessive trait. So there's two possibilities of a genotype here for someone to have free earlobes, because free earlobes are dominant. They could either have the genotype capital F, which would be homozygous dominant, or they could have one capital F, one lowercase f, which would be heterozygous, meaning they have one of each size letter, but they're going to express the dominant trait. So their phenotype, what they actually show, is free earlobes if they have these combinations. For the lower one, the genotype is for recessive would be homozygous recessive. It has to be in order to show the earlobes that are attached. So it would be little f, and the phenotype would be earlobes attached in order to express that. Okay, so now that we've reviewed the terms from 10th grade, let's go into a little bit more about genetics or inheritance. So we got to start the story, and it usually does, is with Mendel. So Mendel was worked at a monastery, and he was known for his genetic experiments he did with pea plants. Before Mendel, though, there was this theory of preformation, which is from the 1700s, and it's the idea that an exact miniature replica of a parent existed inside their sperm or their egg already. So there was no genetic recombination happening. It was more an asexual idea that they were going to be exactly the same genes as their parents. And that was called the theory of preformation. And that's what people were basically believing until Mendel started to create data and evidence about ways that things are inherited or passed on. So we often say he discovered the principles of inheritance, the things that we can go on and believe about how plant or organisms that reproduce sexually, how their genetics work and how certain traits are seen and why certain traits aren't anymore. So a little history about Mendel is that he grew up in a small farm in Austria. He went to the University of Vienna, so we're in the mid-1800s here. He lived in the monastery. He was a monk from 1857. He bred garden peas to study inheritance. And why did he use pea plants? It's because they can grow really fast, and they had some visible traits that made it very easy to identify what was being passed on or not. So his contributions some of the main contributions were alleles that were, he made us understand that alleles were responsible for characteristics that we were seeing or expressing. And then he considered probability of different combinations of offspring. So we could predict what an offspring was going to be. Now, this may not have been so much with humans, but with plants and agriculture and different animals, you could breed and kind of try these things out in experiments. So with pea plants, like I said before, they're distinct traits, and they have a large flower, which allows for easy manipulation of the crosses. So he was actually crossing the traits for them. The plants weren't doing this on their own. He was kind of forcing the idea of making hybrid crosses. But here are some of the traits you can see in plants. Distinctly, their seed shape, so it would be smooth or bumpy. Seed color, yellow or green. Flower color, pink or white. Pod shape, so it would be smooth or rigid. And then pod color, green or yellow. Also, there was tall versus dwarf size plants. And the axial, the way that they grew, this one was considered axial, or this one would be terminal. So you can see this one has multiple buds coming off the axis, where this one had one. So just so you understand, before I talk about what he did to cross-pollinate them, you need to understand the anatomy of a flower. So on this flower, it's showing both female and male traits. Some plants have both, some don't. This pea plant particularly does. So you see, this is the female anatomy right here. So this is called a carpel. This is the ovary. This is the ovule. So this is where eggs are going to be forming in the sense of a female plant, or when we think of a female human, ovaries, right? This is called a style, and this is the stigma. So this is where pollen needs to enter and come down into this tube, into the ovaries. And this is the petals of the plant here. The stamen is the male anatomy of the plant. So these are called anthers, and these are filaments. The anthers are what the pollen sits on top of. So basically, if you can get some of this pollen and rub it here onto the stigma, and it attaches there, some of that pollen will fall inside here into the ovary, and it'll fertilize the flower. So here you can see what he did. So he removed the stamens from the purple flower. So he was dealing with a purple flower and a white flower. The stamen, remember, is the male organ. So he's basically removing the pollen, and then he would brush some of the pollen. He transferred some of the pollen and he removed the pollen from this purple plant because he didn't want, since this flower or this pea plant has both the male and female anatomy, he didn't want this pollen to enter the female carpel. So he cut off the purple flower's stamen, and then he took pollen from the white flower and brushed it onto the carpel of the purple flower. So he's cross-pollinating. A pollinated carpel matured into a pod, and then he planted these seeds and could see what type of flowers bloomed from the first generation. In this case, they all ended up still being purple, even though he used the pollen from a white flower, which the white flower is normally recessive and the purple flower is dominant. So at this point, he would need to do the same thing with this group of plants, plant them again and see if in the second generation any of the white flowers would pop up, because most likely a lot of these flowers are heterozygous. So when we also do Punnett squares, or when we do pedigrees, we use this terminology, so it's good to know. The P stands for the parental generation. This is the first one we're going to cross. So in this picture here, these would be the two parent generations, right? And then this is going to be the F1, which is the first generation of offspring. So what the F stands for is filial, okay, so like a child, so first filial generation. And then F2 would be the next generation, the second filial generation. But you just need to remember P, F1, and F2. These are some of the different crosses that Mendel did, so looking between purple and white flower plants. And then here's the ratio of how often he got the traits to show up. So flower position, seed color, seed shape, pod color, and stem length. All right, so one of the skills we're going to work on today is Punnett squares. So just as a reminder, there are two types of Punnett squares. There's a monohybrid cross, which mono means one, so you're only looking at one gene, one trait. There's something called a dihybrid cross, is where you'd be looking at two traits. A monohybrid cross only needs a 4-box Punnett square, where a dihybrid cross is going to need 16. So what you're going to look at here is first evaluating the gene for yellow peas, is this example. So the parental generation, the sperm is from a YY individual, so it's dominant homozygous, and the egg is from a recessive homozygous, so 2 lowercase y's. So we're going to put one parent on this side and the other parent on this side, and we're going to do the cross. So remember, a trick that I've always taught students to do is to, under the uppercase letter, to draw a line when the letters look very similar, uppercase and lowercase, so you don't get confused. So this box has one uppercase in it and one lowercase, so this will be a heterozygous combination. And same with this one, same with this one, and same with this one. So all 4 possibilities are heterozygous. So this would be 100% heterozygous genotype. And it'll be 100% expressing, though, the yellow trait, since the capital Y represented yellow, because they have at least one capital letter. When you hear the word test cross, this means you're testing a suspected homozygote by crossing it with a known homozygous recessive. So if we know something has the recessive trait, like this white flower, it cannot be white unless it had both recessive alleles. So we call this homozygous recessive. So you would take this and cross it with a flower that you are unsure about. You're trying to figure out if it's homozygous recessive or homozygous dominant or heterozygous. So you're trying to figure that out. It's suspected homozygote. So what you would do, these are the two possible combinations. So if it was homozygous dominant, these would be the possible offspring you would get. If it was homozygous, or if it was heterozygous, these would be the potential you would get. So if you were to cross this flower with a white flower and you ended up getting some white, that would mean you would know for sure this was a heterozygous. If you were to actually plant these pea plants after cross-pollinating them. If you were to get this combination where they were all purple, you would still be unsure. All right. So some traits, though, are codominant, and codominant traits mean that they are both dominant, that they both want to be expressed, and that there is no recessive trait that's going to be, there is still a possible recessive trait, but the codominant traits mean they both are expressed, so they kind of create this new mixture. So for example, some alleles, so the way we would define this is some alleles are equally strong and neither masked by the other. So that means neither is hidden, neither is dominated. So when both alleles are present, they are both expressed in the phenotype. The hybrid is a blend of both alleles. So one example is the ABO blood type, because there is A blood type, B blood type, and AB blood type. So the AB would be that codominant version, and then O is the recessive. Sickle cell anemia is another example of a codominant trait. Roan horse color, which is kind of like a red and white speckled color on the horse, usually on its rear. And this flower here, some flower colors also have codominant traits, which give them this speckled look as well. So let's use the example when we're talking about codominance. We also can use it to talk about multiple alleles. So sometimes some traits are not as simple as just being homozygous, or dominant or recessive. Sometimes they are more complicated than that. So if you remember when we were in 10th grade and you guys had to flip a coin and draw for certain traits, and you had to draw your own child's face, some of the traits like skin color involved 8 different alleles. So you had to flip 4 times to figure out which allele you contributed, the homozygous or recessive, for 4 of them, and then your partner did the other 4. So some traits have multiple alleles involved in creating what we see in the phenotype. So the ABO blood types are an example of multiple alleles. So this is a picture of a red blood cell. What's interesting is that some cultures consider blood types so important. It's not just a genetic trait that you inherit, it's also your personality, and they use it sometimes for dating and relationships and personalities. So I know when I lived in Korea, I was asked often what my blood type was, and this kind of went with what my personality would be. So I'm type A, and this is a Japanese example, so it's very common in Japan as well. And so it seems like a weird question maybe when you go on a date with someone, but they would ask you what your blood type is, because that might give them a little bit of an idea of your personality traits. But back to more scientific looking things. Let's just review what the blood cell is for, first of all. So the red blood cell is there to transport oxygen. That's its main goal throughout your whole body. White blood cells are going to help fight infections. They also are traveling within your body. So you have the red and white blood cells. Platelets, which help blood clot, so that's in your red blood cells. And then plasma, which is the medium that carries these cells. So the plasma is what everything's traveling in. So in this picture here, you have the red blood cells, the white blood cells, the platelets, and then this drop of this liquid is the plasma that it's all moving in. So just as a review of what we're talking about when I say ABO blood types. So each blood type has a specific antigen. So remember, antigens are the surface markers on your cells, in this case, red blood cells. And this is your blood cells like ID system. So the antigen is a marker that is going to be recognized by your body, and it helps it figure out what it shouldn't be accepting, what shouldn't be there. So you're all going to have, if you have the antigen for blood type A, which in this case is these little green spikes here, you're also going to have the antibody for B, so the anti-B antibody. So proteins produced by white blood cells is what that would be, and it would recognize certain antigens and it triggers an immune response. So that shape will know, that antibody's shape or its active site will know that for some reason the type B blood has been put inside your body and it will attack it. So what is special though about it genetically? So what is special about the ABO blood grouping system? It's multiple alleles, so when we talk about it, this is how you have to write it. You can't write A, B, okay? You have to use the capital letter I with an A on it because this is dominant for A. Capital letter I with a B on it represents the B blood type. So remember, they're co-dominant, so they neither wants to mask each other. They're both if they're present. And then you have little I, which is going to represent the O blood type, the recessive trait. So co-dominant, so they're multiple alleles and they're co-dominant. So both alleles are expressed sometimes. This is the co-dominant allele if you have capital IA, capital IB, and the little I is the recessive. So if you look here, if you're going to have type A blood, that means you're either homozygous dominant or you are heterozygous. So the protein on your blood, the antigen is going to be the marker A, and then the antibody will be anti-B. For type B blood, you are either homozygous dominant here or heterozygous with the one capital B and a lowercase I. This would be type B blood. You're going to have the antigen B, and then you have the anti-A antibody. This is the co-dominant version, capital IA, capital IB. That means you're type AB blood, and you have both antigens, so that means you have no antibodies. So if you have type AB blood, that means you are the universal taker. You can take any type of blood. Type O blood is going to have little I on it, so they're type O, and they don't have any antigen markers on their blood. So, and they are going to have the antibodies for both because their blood cells don't have markers on it. So that means they are the universal giver, but they cannot receive any other blood type besides their own. So if you want to look at it from the antigen perspective, so here is group A. This is their antigen, and this would be their anti-B, which notice it's the same shape here, and it'll recognize the group B's antigen. And here's the anti-A body. And then here's the group AB, and then here's group O, and they're going to have both antibodies. All right, so what's the problem with what happens when an antigen meets a specific antibody? And we kind of went over this in the circulatory system, but we call this agglutination. And this is when basically the surface antigens perceive that it's the wrong, or the antibodies perceive it's the wrong type of blood, and they're going to attack it. And this creates agglutination, which is basically a clumping that's going to occur. And then they're going to lyse those blood cells. So you can see it happening in this picture here. It's clumping together, and eventually it'll break and lyse it down, so it'll kill that blood. So if you get a transfusion from blood that's not matching your body, this is what your body is going to try to do to it. And then you won't have enough blood to carry oxygen around your body. So let's do a blood type Punnett square. So I'm going to have you guys pause for us. Well, you're going to finish the video, write down, do this problem on your notes, and then you're going to submit your notes on ManageBack. So first, just to kind of go through it, I want you to do a blood type Punnett square. So you're going to use 4 squares for the monohybrid cross. Remember, these are the blood types right here. Okay. So, and the problem is a woman with blood type AB marries a man with blood type O. What are the possible blood types and genotypes of their children? So, when you do the Punnett square, remember normally there's going to be a capital I on each of these, anywhere where there's a big A. So just keep that in mind as you do your Punnett square. So please go ahead and try to do the Punnett square and figure out the possible blood types and genotypes of the children, and we're going to go over that in class. I'm also going to be assigning you a worksheet of other Punnett squares to do, and that'll be due by next class. Let me know if you have any questions, and I hope that was helpful.